Hello friends, can you hear me? We are live on the YouTube. I'll just take a different audio and see if it is working. Not working here. Can you hear me? Everyone, can you hear me? Is the audio clear? I'll also check at my end. Can everyone see me and can you hear me? Send in your messages. Can you see me? Can you hear me? Am I audible? Am I audible? Can you see me? Can you hear me? Am I audible? Check at my end. Can everyone see me and can you hear me? Send in your messages. Can you see me? Can you hear me? Am I audible? Am I audible? Can you see me? Can you hear me? Okay. This looks perfectly fine. This looks perfectly fine. That's great. That's great. So I'll put it over here. All right. Let's make it full screen. All right. So we are continuing with our session. We are continuing with our session where we had started with the second important group of inborn error of disorder, and it is related to the phenylalanine tyrosine complex. All right. So phenylalanine normally converted to tyrosine by the enzyme phenylalanine hydroxylase. It is converted to tyrosine by the enzyme phenylalanine hydroxylase. In some individuals, the phenylalanine hydroxylase may not be there, or it is greatly decreased in amount, giving rise to the condition known as the classical phenylketonuria, which is nowadays known as phenylketonuria type 1. Deficiency of phenylalanine hydroxylase, please note, deficiency of phenylalanine hydroxylase gives rise to the phenylketonuria type 1, also known as the classical PKU. For this reaction, we also require the coenzyme. The coenzyme required is the tetrahydrobapterin, which gets converted to the dihydrobapterin. The dihydrobapterin has to be recycled into the tetrahydrobapterin, the main enzyme, not the only enzyme, but the main enzyme in this case is the dihydropteridine reductase. In some individuals, there can be defect either in the biosynthesis, requires two enzymes, or in the recycling, which also requires two enzymes. There may be defect giving rise to again the phenylketonuria, but this phenylketonuria is malignant type, nowadays further subdivided into type 2, type 3, type 4, and type 5. So basically, in the current scenario, PKU will be of 5 types. Type 1, which is due to deficiency of enzyme the phenylalanine hydroxylase. Type 2, 3, related to the biosynthesis, where the defect is in biosynthesis. These are type 2 and 3. Type 4 and 5 related to the recycling. Type 4 and 5 related to the recycling. We'll talk about the details of the disorder very soon, but let's first complete the metabolism and the associated disorders, and then we'll write the details. The tyrosine is then converted into the hydroxyphenyl pyruvate. The enzyme required here is the tyrosine aminotransferase. The enzyme required here is the tyrosine aminotransferase. Deficiency of this enzyme gives rise to the tyrosinemia type 2. The tyrosinemia type 2 has some other names also, I'll tell them to you later, but the more important name is the tyrosinemia type 2. So phenylalanine to tyrosine, tyrosine to hydroxyphenyl pyruvate. Hydroxyphenyl pyruvate is then converted into homogentisate by the enzyme hydroxyphenyl pyruvate oxidase. The enzyme which is working here is the hydroxyphenyl pyruvate oxidase. Now here we can have two scenarios. Number one, the enzyme is deficient. Enzyme is not there, it is less in amount. In that case, we have the neonatal tyrosinemia, also known as the tyrosinemia type 3. It is the mildest type of tyrosinemia, the neonatal tyrosinemia or the tyrosinemia type 3. In other individuals, the hydroxyphenyl pyruvate may be defective. See, there are two things. One is deficient, enzyme is not there. In other individuals, enzyme is there but it is not working properly, so it is the defective enzyme. In this case, the disorder that we'll get to see is the Hawkinsinuria. I'll tell you about this, don't worry. The disorder seen in this case is the Hawkinsinuria. If the enzyme is not working properly, if it is defective, then the condition that we get is the Hawkinsinuria. Then homogentisate has to be converted into fumaryl acetoacetate. The enzyme required here is the homogentisate oxidase. Deficiency of this enzyme gives rise to the L-captonuria. L-captonuria is a quite commonly asked question in your exam, so you should remember this. L-captonuria. L-captonuria. Okay. Subsequently, finally, the fumaryl acetoacetate is converted to fumarate and acetoacetate. Fumaryl acetoacetate converted to fumarate and acetoacetate. The enzyme required here is the fumaryl acetoacetate hydrolase. Deficiency of this enzyme gives rise to the most important tyrosinemia type 1. Tyrosinemia type 1 is the most severe type of tyrosinemia among all the tyrosinemias, the tyrosinemia type 1. So let's quickly do the recap of the disorders and then go into each of the disorders one by one. Okay, phenylalanine tyrosine enzyme phenylalanine hydroxylase disorder is phenylketonuria classical type, also known as PKU type 1. Coenzyme required tetrahydrobiopterin, which is converted to dihydrobiopterin, which has to be recycled. Either the biosynthesis or the recycling may be defective, giving rise to the phenylketonuria malignant type, also subdivided as type 2, 3, 4, and 5. Tyrosinemia is then converted into the hydroxyphenyl pyruvate by the enzyme tyrosine aminotransferase. Deficiency gives rise to the tyrosinemia type 2. Hydroxyphenyl pyruvate is then converted into the homogentisate. The enzyme required is the hydroxyphenyl pyruvate oxidase. Deficiency gives rise to the disorder neonatal tyrosinemia, also known as tyrosinemia type 3. If the enzyme is defective, then we get the condition known as Hawkinsinuria. Homogentisate acted upon by the enzyme homogentisate oxidase gives rise to fumarate acetoacetate. If the enzyme is deficient, then we get the L-captonuria. And finally, the fumarate acetoacetate converted to fumarate and acetoacetate by the fumarate acetoacetate hydrolase. Deficiency of this enzyme will give to the most severe type of tyrosinemia, tyrosinemia type 1. Now that we have listed the disorders, let's look at the disorders one by one. What are the different points that you must know about these disorders? Starting with PKU. PKU is a fairly common question. So what I want from you is tell me what is the most important presenting feature of PKU for which we are worried. Most important presenting feature of PKU which is of clinical concern. I'm waiting for answers. Most important clinical feature of PKU. PKU is one of the most important causes for mental retardation. And another problem is even after you diagnose and you start the treatment, even after you diagnose and start the treatment, the mental retardation is not reverted. Irreversible mental retardation is seen. So you have to diagnose it at birth and you have to start the treatment right at the time of birth. Okay, So what are the points you need to know here? We already mentioned the PKU now labeled as a type 1 and 2, 3, 4, and 5. In type 1, one of the important classical findings is the mousy odor of urine due to phenyl acetate. See, these are two different aspects of the question you should know. What is the odor? The odor will describe as mousy. Another term which can be used here is musty, mousy or musty odor of urine. The second part of the question would be what is the analyte responsible for this odor, and that is the phenyl acetate. That is the phenyl acetate. So mousy or musty odor of urine. This odor may also be present in sweat, not only in urine. It can be present in the sweat also, and the inheritance pattern is autosomal recessive. The inheritance pattern is autosomal recessive. Now here I'll mention two three things which are additional points. It is the third most common cause of you can say inherited mental retardation. Third most common cause of mental retardation, behind the Fragile X syndrome, the Down syndrome, the Fragile X syndrome, and then we have the PKU as the important cause for mental retardation. So it is fairly important and likely to land up in exam. Okay. Second, how do you manage the patients of PKU? How do you prevent the mental retardation from occurring? We prevent the mental retardation from occurring by not providing the phenyl aniline, which is not getting metabolized. See, I'll go back to that flow chart that we had seen. So here, if the phenylalanine is not converted to tyrosine, then it gets metabolized by alternate route, whereby we are forming the phenyl lactate, the phenyl acetate, and these are the molecules or the compounds which are responsible for the toxicity of the disorder. So what we have to do so that the mental retardation does not occur, these compounds should not be formed. To prevent these compounds from being formed, we have to decrease the concentration of phenylalanine only. So the management in these patients is very simple and straightforward, which we have practiced previously also, and that is dietary restriction. You have to restrict the phenylalanine from diet. Dietary restriction of phenylalanine is practiced in patients of PKU. The dietary restriction of phenylalanine is practiced in patients of PKU. Okay, let's go back to our table on PKU. Okay, so dietary restriction is practiced in patients of PKU. Now, just a side note, how are you going to advise the patient to follow the dietary restriction? You tell them, okay, you are not supposed to take phenylalanine. What will the patient tell you? Sir, what do you mean by not take phenylalanine? He doesn't know what is phenylalanine. He doesn't know in which food stuff the phenylalanine is there. For that matter, do you know which food stuff should not be consumed, which are rich in phenylalanine? The problem with phenylalanine is that it is one of the most common amino acids present in the food. So we tell them no protein from outside. We give them special formulations which contain all the essential amino acids. So if you talk about management, you have to give special formulations. It contains all essential amino acid minus phenylalanine plus tyrosine. See, in these patients, the tyrosine becomes an essential amino acid because normally the tyrosine is formed from phenylalanine, but that step is getting blocked in these patients. So here the tyrosine becomes an essential amino acid. This is a very important point to keep in mind. The tyrosine in these patients becomes an essential amino acid. So we give them special formulation which contain all the essential amino acid without phenylalanine, but tyrosine is added. Second part of this aspect is Till what time the dietary restriction of phenylalanine has to be followed? Initially, different ages were advised: adolescent, adulthood, maturity, puberty. But now, the current advice is that you have to give the restriction, the dietary restriction, throughout life. The dietary restriction must be followed throughout life. The dietary restriction has to be followed throughout life. Okay. Then the second part, where we said the malignant PKU is now subdivided into type 2, 3, 4, and 5. Okay, 2 and 3 is due to regeneration, not due to biosynthesis. 2 and 3 due to defect in regeneration. 4 and 5 is due to defect in biosynthesis. In the diagram, I had written it wrong. The 2 and 3 is due to defect in regeneration. 4 and 5 is due to defect in biosynthesis. Both the process requiring two enzymes each, and therefore we have two subtypes for each of the scenarios. Will not ask you about the malignant PKU. Just remember that the problem is there. The reason is because of the tetrahydrobaptarin. Tetrahydrobaptarin is also required for conversion of tyrosine to dopa. I had shown you yesterday the tyrosine to dopa requires the tetrahydrobaptarin. So if tetrahydrobaptarin is not there, this will also not be formed. If this is not formed, the catecholamines cannot be formed, and therefore we have more severe features in the malignant PKU because the catecholamines are also not being formed. In classical PKU, there is no problem in the synthesis of the catecholamines. Okay, moving on to the tyrosinemia type 1. The tyrosinemia type 1 is the most severe type of tyrosinemia, also known as the hepatorenal tyrosinemia. The other name for this is tyrosinosis. This question was asked in the 2018 NEET exam, where they had mentioned the enzyme deficiency was asked for tyrosinosis. Now, most of the students did not know that tyrosinosis is the name for tyrosinemia type 1. The other name is the hepatorenal. This is based on the organs which are involved in the tyrosinemia type 1. In hepatorenal tyrosinemia, the two organs involved are the kidney and the liver. We already seen defective enzyme is the last enzyme, fumarate acetoacetate hydrolase, and the presenting features are related to the progressive renal disease and the renal tubular dysfunction, which presents with rickets. Recently, a drug has been launched a decade back, the NTBC or nitisinone. The NTBC or nitisinone has been launched a decade back, which is used for the management of patient of tyrosinemia. My question is, what is the urinary odor in patients of tyrosinemia type 1? This odor is not seen in tyrosinemia type 2 and 3. If at all it is there, it is very mild, but it is very prominent in patients of tyrosinemia type 1 or tyrosinosis. What is the urinary odor in patients of hereditary tyrosinemia type 1, also known as the hepatorenal tyrosinemia, also known as the tyrosinosis? What is the urinary odor in these patients? You have to tell me the urinary odor in patients of tyrosinemia. The answer is boiled cabbage odor. The boiled cabbage odor. Very good, Misala. The answer is cabbage odor. That's more, to be more precise, it is the boiled cabbage odor. Okay, so this is the tyrosinemia type 1, also known as the hepatorenal tyrosinemia or the uh, tyrosinosis. The tyrosinosis. Another important feature in these patients can be the calculi, renal calculi. where we have the tyrosine stones. Tyrosine stones can be seen in these patients because of the precipitation of tyrosine in the renal calculi. In the renal calculi, the precipitation of the tyrosine gives rise to the calculi. The tyrosine stones are formed. Okay, the tyrosinemia type 2 I told you has an additional name also. That name is the Richner-Hanhart syndrome. This is a very important name, very commonly asked. The Richner-Hanhart syndrome, also known as the tyrosinemia type 2. Due to the involvement of features, it is called the oculocutaneous tyrosinemia. It is also known as the oculocutaneous tyrosinemia. It is not as severe as tyrosinemia type 1. What we find here is the keratitis, the skin involvement, sorry, the eye involvement, and the blistering lesions of the palms and soles. So keratitis is the ocular part, and the blistering lesions is the cutaneous part of the name. The oculocutaneous tyrosinemia, because of the presentation with keratitis and blistering lesion of the palms and the soles, that's tyrosinemia type 2. And then we come to the neonatal tyrosinemia, also known as tyrosinemia type 3. I told you it is the mildest form. In most of the patients it is asymptomatic. In most of the patients it is asymptomatic, or rarely it can be associated with mental retardation. Mental retardation is rarely seen, only in a very low percentage of patients. That's the tyrosinemia type 3. I told you for the same enzyme, for the hydroxyphenyl pyruvate oxidase, if deficiency is there, we get the tyrosinemia type 3. If defective enzyme is there, if there is defective enzyme, then we get another disorder. The name of the disorder was Hawkinsinuria. All right, this is because of incomplete metabolism of the target. The target is the hydroxyphenyl pyruvate. The target is hydroxyphenyl pyruvate. The intermediate which is formed is called Hawkinson. The intermediate which is formed is the Hawkinson. That's why the condition is known as Hawkinsinuria. Just like the tyrosinemia type 3, a large number of patients will be asymptomatic, but rarely we may find the failure to thrive. The failure to thrive is associated with metabolic acidosis. The failure to thrive is associated with metabolic acidosis. What is the urinary odor in these patients? In patients of Hawkinsinuria, we have a distinctive urinary odor, which is a very common odor. Most of us have encountered this odor. So, what is the urinary odor in patients of Hawkinsinuria? The urinary odor in these patients is chlorine like odor, because chlorine is used to purify the swimming pool water. The other name is the swimming pool water odor. It has the same odor that of the chlorine or the bleaching powder. So the swimming pool water odor. Okay, and now we come to the last disorder in the list. And that is the alkaptonuria. Alkaptonuria is another important disorder which is commonly asked. Defective enzyme we already seen is the homogentisate oxidase. The urine becomes black on standing. This is the primary complaint. The urine becomes black on standing. This is the primary complaint. Now obviously the newborn cannot go to the washroom to use the urinal, so he'll pass the urine in the diapers, and the diapers will throw the parents will throw the diaper in the dustbin. The next day when they're giving it to the garbage person, they find that the diapers are black. That is when they become very very anxious. The parents complain of black diapers. They complain of black diapers. This is due to oxidation of homogenticate to what is known as L-captone. L-captone is a black pigment. So the homogeneous is getting oxidized. It is getting oxidized in presence of oxygen to L-captone. So when the urine is passed, it will be clear. If you allow it to stand for some time, you expose it to the air, it will get oxidized and it will form the L-captone, that is the black pigment. Besides this, there are no other sign and symptom in the children right in the second decade, right up to the third decade. By the end of third decade, they start presenting with another set of symptoms, and that is threefold. The erosive arthritis, which normally occurs in the 60s and 70s, the back pain due to fibrosis of the back, and ochronosis. Ochronosis is the same thing, the black pigmentation of the skin and cartilage. The black pigmentation of the skin and cartilage is known as ochronosis. Another question which can be asked here: what is the drug used for the management of patient of L-captonuria? The drug used for the management of patients of L-captonuria. Okay. Any answers? Any answers? Betaine. No. The correct answer is we have already seen the drug which is being given, and the name of the drug is anti-BC or nitisinone. The same drug which is given for the thalassemia type 1 is also useful for L-captonuria. The same drug the given for the thalassemia type 1 is useful for the patients of L-captonuria. So the same drug will be used here also. So this is the second very important cluster of the inborn error of metabolism. The first cluster was the urea cycle disorder. The second cluster is the phenylalanine tyrosine metabolism defect. Now, besides this, we have a group of disorders called the branched chain organic aciduria or acidemia. If you examine the urine, then aciduria. If you examine the blood, then it is called the acidemia. Now, what you have to remember here, two things. Number one, all the names in this list are related to the defective fatty acid metabolism except one. All the names in this list are related to the defective fatty acid metabolism except one. Okay, and that name is the maple syrup urine disease. That name is the maple syrup urine disease, which is due to the defective metabolism of the amino acid. It is due to the defective metabolism of the amino acid. All right, so we'll take a look at the maple syrup urine disease. In addition, what you should know is a characteristic odor in isovaleric aciduria, and that is the sweaty feet odor. Sweaty feet odor of urine is seen in patients of isovaleric aciduria. Sweaty feet odor seen in the patients of isovaleric aciduria. Let's go to the maple syrup urine disease. Remember, it is the only branch in organic aciduria related to the defect in metabolism of amino acid. All others are related to defect in the metabolism of the fatty acid. The maple syrup urine disease is due to deficiency of an enzyme complex which we have talked about previously, and the name of that complex is the branched chain keto acid dehydrogenase, the enzyme which is similar to PDH. If you recall, when we're talking about PDH, we had mentioned the name of this enzyme, the branched chain keto acid dehydrogenase. The branched chain keto acid dehydrogenase deficiency of this enzyme gives rise to the maple syrup urine disease. So what is happening here? The defect is in metabolism of the branched chain amino acid. Defect is in metabolism of the branched chain amino acid. Which are the branched chain amino acid? If you recall, we had mentioned yesterday valine, leucine, and the isoleucine. These are the branched chain amino acid. So the amino acid, their keto acid, and their hydroxy acid, all of these will increase in blood. If they are increased in blood, they will also be increased in urine. The plasma and urinary levels of leucine, isoleucine, valine, their keto acid and their hydroxy acid are elevated. Both plasma and the urinary levels. Both plasma and the urinary levels. Inheritance pattern is autosomal recessive. The blood marker is elevated allo-isoleucine. The blood marker is the elevated allo-isoleucine. If you find this to be elevated, it will be common in all the patients of MSUD. Now in MSUD also we have some degree of mental retardation, or you can say the neurotoxicity is there. So the question which is asked is, which intermediates will cause the neurotoxicity? The answer is leucine and the ketoisocaproic acid. These are the two molecules which will cause the neurotoxicity. They will cause the neurotoxicity. Okay, the odor of urine in these patients. What is the urinary odor in these patients? Urinary odor. Obviously, some of you will say the maple syrup odor is there, but what does it smell like? The maple syrup, what does it smell like? What can you compare it with? The urinary odor, we describe it as the burnt sugar odor. The burnt sugar can also be called caramelized odor, or you can say caramelized sugar odor. So remember, the burnt sugar odor or the caramelized sugar odor, it is not the taste, it is the odor. All right, the in maple syrup urine disease, the urine does not taste like maple syrup, it smells like maple syrup. It only smells like maple syrup, it does not taste like maple syrup. Very good, it is the burnt sugar odor. Okay, so till now we have seen the various defective metabolism whereby the amino acid or the metabolites are coming in the urine. Now sometimes the metabolism may be normal, but there can be defect in the transport of amino acid whereby the amino acid may be lost in the urine. So we look at some important disorders where the defect is there in the transport of the amino acids. The first in this list is the cystinuria. The first in this list is the cystinuria. What you have to note here, in cystinuria, the four amino acids lost in the urine, remembered by the name COLA, are cysteine, ornithine, lysine, and arginine. This is not a spelling mistake, it is cysteine and not cysteine. Please note that the name mentioned is not a spelling mistake. The amino acid which is lost is cysteine. Cysteine is normally absorbed. The absorption of cysteine is normal. It is not lost. It is normally absorbed. The cystinuria is of two types, the type 1 and the non-type 1. What you remember are the names of the defective genes. Which genes are defective in cystinuria? They are SLC3A1 and the SLC 7A9, SLC3A1 and SLC 7A9. Remember, all of these amino acids are dibasic. The cysteine is monobasic. What is cysteine? Two cysteine joined together will give you the cysteine. So cysteine is dibasic, whereas cysteine is monobasic. Cysteine is monobasic. So that is the first disorder, the defect in amino acid transport of the uh, dibasic amino acid. You can remember them by the mnemonic cola. I'll highlight here. The cola is not the color of the urine in this case. See, there are various conditions where the hemoglobin and the myoglobin and the RBC may come in the urine, where the urine is cola colored. Here, the cola is not for the color. Cola is a mnemonic for the amino acid which are lost in the urine. The urine is normal in appearance. The urine is normal in appearance. It is not the color. The urine is not cola colored. Okay. Second disorder in the list is the Hartnupp's disease. In Hartnupp's disease, the defect in transport of 13 different amino acids has been documented. 13 different amino acids are lost in the urine. So obviously, large number of amino acids are lost. It doesn't make sense to remember the names. What you have to remember is it manifests as tryptophan deficiency. Why? How? Remember, tryptophan is required for synthesis of niacin. 60 milligram will give rise to 1 milligram of niacin. So tryptophan is lost in the urine. If tryptophan is lost in the urine, then the niacin will not be formed. If niacin will not be formed, then we will see the pellagra. So the clinical presentation is that of pellagra. You supplement with either niacin or the tryptophan to alleviate the signs and symptoms. Okay. The defective gene, just like in the previous disorder, is the SLC6A19. SLC belongs to a family of transporters which is known as SL for soluble and C for carrier. Okay, so these are active transporters which will commonly transport the amino acids. Active transporters which will commonly transport the amino acids. Okay, so these are the very important amino acid related disorders which you must know about. Uh, so very frequently the clinical case will come from these questions only. They'll give you one or more features of the disorder. They may give you the clinical presentation of the disorder also along with the biochemical features, and then you will be asked a question related to the disorder. Maybe what is the treatment, what is the management, what is the enzymatic defect. All these questions can be asked related to the disorders. All right, any questions so far? Any questions so far? No questions? There are no questions. If there are no questions, then I will ask questions. If there are no questions, then I will ask questions. Take a look at the question. Question number one. Transfer of an amino group from an amino acid to an alpha keto acid is done by which of the following? Your options are the transaminases, the aminases, the transketolases, and the decarboxylase. Which of the following is the correct answer? Very good. So the correct answer is the transaminase. See, this statement is basically the description of transamination. This statement is basically the description of transamination. Obviously, the enzyme is transaminase, and remember, the transamination is a freely reversible reaction. The amino acid which serves as a carrier of ammonia to liver. The options are glutamine, alanine, methionine, and arginine. Which of the following is the correct answer? Which of the following is the correct answer? The amino acid which serves as a carrier of ammonia from skeletal muscle to the liver. Glutamine is the transporter for all tissues except muscle, whereas muscle, the transport of ammonia is done by alanine. For the muscle, the transport of ammonia is done by alanine. So the correct answer is 2, alanine. Correct answer is 2, that is alanine. Very good. Pooja, Supriya, Lunavat, Simha, Mamta, very good. They are the correct answers. Even Srujan, Murtaza, very good. Akshay, well done. So we come to the next question. Which product of citric acid cycle is used in detoxification of ammonia in the brain? I had shown you this point multiple times, and I am sure you should be able to answer the question. Your options are oxaloacetate, the alpha ketoglutarate, the succinate, and the citrate. Which of the following is a product of citric cycle used in detoxification of ammonia in the brain? Obviously, the correct answer is alpha ketoglutarate. Alpha ketoglutarate will chelate the ammonia and get converted into glutamate. Very good, very good. The correct answer is the alpha ketoglutarate. Alpha ketoglutarate is responsible for detoxification, and its depletion is responsible for the encephalopathy. The depletion of alpha ketoglutarate is responsible for the encephalopathy. As you can see here, straightforward question, single line question. The urea cycle occurs in which of the tissues, whether it is the GIT, spleen, kidney, liver? And as you know, the correct answer is liver. If liver is not there in the option, you can mark the brain. If liver is not there in the option, you can mark the brain. Next question: sources of nitrogen in urea cycle. If you have seen the urea cycle carefully, you should be able to tell us the sources of nitrogen in the urea cycle. Your options are the glutamate and ammonia, the arginine and ammonia, the aspartate and ammonia, and the uric acid. If you had paid attention to the urea cycle, you should be able to give us the answer. The answer is aspartate and ammonia. This aspartate is coming from the recycling of fumarate via the mitochondria. It will combine with the citrulline, and ammonia is used in the first step. Ammonia is used in the first step. Very good. That's the correct answer. Which of the following enzymes is a urea cycle enzyme? Which of the following enzymes is a urea cycle enzyme? Glutamine is primarily used after transport, so it is a part of the transport. Asparaginase not involved. Glutamate dehydrogenase is for deamination as well as transport. So the correct answer is enzyme 3 of the urea cycle. Enzyme 3 of the urea cycle is the arginosuccinate synthetase. Very good. Mesla, Manisha, Pooja, Avinay, Srujan, you have given the correct answer. The arginosuccinate synthetase is the enzyme of glycolysis. Next question: at physiological pH, carboxy terminal of a peptide is, remember we had mentioned physiological pH means 7.4. Carboxy terminal at any pH greater than 3 will be negatively charged. We have seen the carboxy terminal at any pH greater than 3 will be negatively charged. हम्म, क्या चाहिए? एक पिक्चर प्रिंट आउट। कहाँ पिक्चर ले आओ, प्रिंट आउट कर दूंगा मैं। मैंने ड्रॉ नहीं करा, वेट करोगे तुम? हाँ, वेट करके ले आओ, प्रिंट आउट कर दूंगा। Okay. Next, we come to which one of the following can be a homologous substitution for isoleucine in a protein sequence. Now, I told you homologous substitution means similar amino acid as per R side chain. So the first classification that we had seen, based on the R side chain, that is used to describe the homologous and the non-homologous substitution. All right. So when we talk about the homologous substitution of isoleucine, it should be similar to isoleucine. For this, you should know what is isoleucine, what type of amino acid is the isoleucine, and if you recall, it was the branched amino acid. So the replacement must also be the branched amino acid. Which are the branched amino acid? Valine, leucine, isoleucine. So among these, the answer will be valine. Very good, very good, very good. Abhinay, Masala, Mamta, the answer is valine. The amino acid having amino side chain, we had specified this separately. The amino acid having amino side chain, that is why it is also known as the amino acid rather than the amino acid. It is sometimes known as the amino acid. And the answer here is proline. The answer here is proline. If you care to look at the structure is something like this. So this is the amino group, and you can see why amino group, because we only have the NH. We only have the NH. We don't have the NH2. So we call it the amino group, and the molecule is known as the amino acid. Basic amino acids are HLA. HLA. A. So the correct answer will be proline. The correct answer will be proline. The basic amino acid are HLA. So the answer is proline. Proline is not part of the group of basic amino acids. Non-essential amino acid. So here you should know the essential amino acid. Essential amino acids are histidine, methionine, valine, isoleucine, phenylalanine, lysine, leucine, tryptophan, and threonine. In addition, arginine is semi-essential. Arginine is semi-essential. This T is for tryptophan and threonine. So the correct answer is phenylalanine. Phenylalanine can be made from, sorry, tyrosine can be made from phenylalanine. We had seen previously the tyrosine can be made from phenylalanine. So it cannot be the essential amino acid. It is a non-essential amino acid. It is synthesized in the body. It is being synthesized in the body. So the tyrosine, the non-essential amino acid is tyrosine. दूसरा ले आएंगे अभी एक ब्लैक वाला पहन लो दूसरा ले आएंगे ठीक है ब्लैक वाला पहन लो ये दूसरा ठीक है तो अभी ब्लैक वाला भी पहन लो ये बाद में दूसरा ले आएगा जाओ अच्छी तरह को चिपका दूंगा मैं रखो गो okay amino acid with dissociation constant closest to physiological pH. We had mentioned this quite a few times. The amino acid with dissociation constant closest to the physiological pH. This is due to the side chain of imidazole. I had mentioned this imidazole. The imidazole side chain has a dissociation constant close to physiological pH. The imidazole side chain is present in histidine, right? The correct answer is histidine. The correct answer is histidine. It is the only amino acid which has the dissociation constant close to physiological pH, and therefore the histidine is the only amino acid which can act as buffer. So those proteins which have large number of histidine will be able to act as buffer. The proteins having large histidine can act as buffer. For example, hemoglobin, albumin. These are some amino acids having large amount of histidine and therefore they are able to act as good buffers. Amino acid secreted in cystinuria are all except. So O is correct, A is correct, L is correct, histidine is not correct. So the answer is histidine. The answer is histidine. Remember the mnemonic cola for the amino acid lost in the urine. It is not the color of the urine, it is only the mnemonic for the amino acid lost in the urine. Which of the following is not synthesized from tyrosine? Norepinephrine comes from tyrosine, tyrosine comes from tyrosine, dopamine comes from tyrosine, melatonin comes from tryptophan. Melatonin comes from tryptophan. Okay. For the previous question, nobody has given the correct answer. Now the correct answer is coming. Now the correct answer is coming. The melatonin will come from the tryptophan. Okay. Next, let's look at this question. Which amino acid does not show post-translational modification? So basically, the Selenocysteine has the genetic code, so post translational modification is not required. Post translational modification not required. For all others you will require the post translational modification. Very good, Masala and Murtaza. The correct answer is the selenocysteine, definitely. In phenylketonuria, the main aim for first line therapy is to replace the deficient product, to reduce the substrate of the enzyme, to change the conformation of enzyme, supplementation of the limiting amino acid. So, I'll just quickly mention what we are doing in the first line therapy. What is the first line therapy? I told you first line therapy is dietary management. Why are we doing the dietary management? We want to decrease the level of phenylalanine so that the alternate metabolic products are not formed. So what we are doing, we are reducing the substrate for the enzyme of the alternate pathway. By doing the dietary restriction, we are reducing the phenylalanine available for the alternate pathway. The mode of action of phenylbutyrate in urea cycle defect is, remember, phenylbutyrate will first get converted to phenyl acetate. The phenyl acetate will then combine with glutamine to form the phenyl acetyl glutamine, thereby removing nitrogen by an alternate route. By removing nitrogen by alternate route, we had mentioned this yesterday, how to manage the patients of the urea cycle. So here we have completed the main topics being asked in the exam from amino acids. If you have any diet, if you have any question, we will take that. Else we'll take a short break for 10 minutes. You can have a cup of tea or coffee, and then we'll start with the topic of proteins. Here we'll talk about collagen also, and time permitting, we'll also be talking about the heme metabolism. So if you have any questions, just let me know. Any questions? Any questions? If you have, just let me know. Else we'll take a short break of 10 minutes, short break of 10 minutes, and then we will continue with the proteins. Any questions?